Hi, everyone. Thanks for joining a new lecture for CPH 290. Today, it's a great pleasure to have Kajsa Kvist. So Kajsa is Scientific Vice President at Novo Nordisk. She is an experienced professional in the fields of biostatistics and scientific leadership. She previously held several senior roles, including Senior Scientific Director and Senior Digital Innovation Director. Prior to Novo, she worked as an actuary at PFA Pension, as well as a biostatistician at Lundbeck. And Kajsa has also contributed to academia as an external associate professor and assistant professor and a postdoc at the University of Copenhagen, where she earned her PhD. Thank you so much for Kajsa for joining us today, despite the time zone differences, and I'll hand it over to you.
Okay, I'm on mute. Yes, thank you so much for inviting me for this opportunity to talk about what we work on in the field of AI and real-world evidence at Novo Nordisk. As Ahmed so kindly said, so my name is Kajsa and I've worked here for quite a long time. I have a background as a statistician, but it is some time ago since I did any of the actual work. So all of what I'm presenting is courtesy of other people's great work in my team. And I put together quite a long presentation, so we can jump through some of it if you have questions, or if you would rather hear other examples. I thought I would start by just introducing the Novo Nordisk R&D organization, what we work with, and what kind of organization we are. Ahmed said that this was the only pharma presentation, so possibly it's of interest. And then I would just run through a number of examples of how we use AI primarily in the R&D organization, but also how we use real-world evidence. And the reason why I point out real-world evidence is because this is a field that requires somewhat advanced analytics, and maybe one could even say borderline AI at times. So I brought a number of examples there. So first, just a little bit about Novo Nordisk. We are a global healthcare company. We have just had a 100th birthday, and we have a headquarter in Denmark, in Scandinavia, Europe. And we have a purpose centered around chronic diseases. So in global healthcare, you talk about chronic diseases and infectious diseases and oncology, but we focus on chronic diseases, and our heritage is built in diabetes. We supply nearly 50% of the world's insulin. We have affiliates in 80 countries, and we employ almost 70,000 people. We have R&D centers in China, Denmark, India, UK, and several places in the US. And we have production sites in Denmark, Brazil, China, France, and the US. And I think this slide is actually a little old, because at our recent earnings call, we came up to serving about 43 million patients worldwide living with diabetes and obesity. And our focus areas is cardiovascular disease, rare disease, hemophilia and growth disorders, diabetes, and obesity. And as you can see here, we are centered in that little blue dot on top of Europe. But we are also heavily presented in North America, in the US, in South America, and in Asia. So just a little bit about our value chain. So we start with the ideation in R&D, where we, based on promising target discoveries, we investigate if molecules have the promised efficacy and do some preclinical testing and clinical testing. Later on, if it seems promising enough, then we move into human testing in clinical. And then if it obtains a positive risk benefit through the very long clinical testing, then we go to product registration, which is engagement with regulatory. Then we engage with the medical community to talk about this new treatment that becomes available for the therapy areas. And we advise on safe and correct use. And then we finally have access to medicine, in which case the commercials and strategy operations step in and handle the communication. So I'm centered very much in the early R&D phase, where the products are not on the market for patients yet. The Novo Nordisk Development Organization, it has an ambition about being the best in class to drive innovative treatments to patients. And if you look at global healthcare, then you have some that's very R&D driven, and then you have some that are more sort of, once the patent treatment goes off patent, then they upscale and primary sort of make treatments that's already been invented otherwise, but we drive new treatments to the market. We have 5,000 employees across the world. The majority of them works in development, medical and regulatory affairs. We have a pipeline that looks at 35 different treatments spanning first in human dose, so the very first time we try to see how it works in humans, and then all the way to it goes to the market. The investigation in humans goes on in clinical trials, and just last year we ran over 168 clinical trials investigating the treatments in approximately 40,000 people across the world. Our strategy is that we base very much our heritage in diabetes, where your blood sugar is typically very high. Then we have, in the last decade or so, branched from diabetes into obesity. These two diseases have a bunch of comorbidities that encaptures renal disease, cardiovascular disease, liver diseases, and also increases the risk of neurodegenerative diseases. And as a sort of other branch that we have also have a long legacy in is rare blood disorders, so hemophilia, and rare red endocrine disorders. So this could be growth disorders. We focus on innovation, bringing new treatments, new treatment modalities, or new proteins to the market. We do this with internal innovation, but we have a heritage of partnering with academia to bring new treatments to the market. So we were based actually on bringing insulin to the European market based on a collaboration with University of Toronto back in 1923. And much of our recent success is based on a collaboration with University of Copenhagen, who was one of the two institutions in the world that discovered GLP-1. Then our drugs are largely based on proteins and peptides, but we also look into antibodies and small molecules. We have cell therapy, so stem cells, for example. We are slowly working into gene therapy and nucleic acids. Our products usually come by injection because proteins are large molecules, so it's difficult to wrap them into a pill that you can swallow. So you have to inject it, but we have some oral delivery, which is really based on engineering, innovation, and ingenuity. So as I said, I was based in development, where we, in order to bring a product to the market, you have to show that it's safe and that it does what we intended to do, which means that we have to test it in humans and supervise how they fare. So we do clinical trial activity across the world. And as I mentioned in the first one, lately we have looked into 40,000 people going through more than 168 clinical trials, which means that we have more than 6,000 sites across the world where patients go through the sites. And for now, we have 9 different therapy areas, but within each therapy area, so for example, within diabetes, we can have more than one treatment. So we can have human insulin, which is a very short-lived, the half-life is very short, an old insulin, or we can have very novel insulins that have a very long half-life, so you only need to inject it once a day. But we also have GLP-1 that lowers your blood sugar and other treatments for diabetes. And as you can see, our clinical trial activity across the 4 phases. So usually you say phase 1, that's the first time you try it out on humans, and that's a very restricted area where usually you have patients coming in to very few sites, maybe just one site, and they are supervised from you give them the injection, and then they have to stay at that site for a week or 2 weeks to see what goes on. So there is medical staff ready if something happens that was not supposed to happen. Fortunately, that happens very, very, very rarely because it's usually extensively tested before we try it in humans, or it's a known biology. Then you have phase 2, which is typically called sort of dose finding, where you figure out what would be the optimal dose to ensure safety, but also get the maximum efficacy. And then once you have found the dose, then you move on to the large phase 3 trials, where you really try to get the clinical confirmation that it works and it's safe. And based on your phase 1, 2, 3, you go to regulatory and get it approved, hopefully. And if it's approved, then maybe you try to expand the use of the drug, and then you go into phase 4. So for example, you can say this treatment is really works well for diabetes, but maybe it also has a possibility in liver disease, and then you go and expand your the client, the patient base. But it can also be that you start with, of course, adults, and then you would like also to get a pediatric label, and then in phase 4 you do a study in children or teenagers, or other vulnerable patient populations. You usually don't do that until the safety is established in the more general population. And then I thought I would just touch upon the therapy areas that we are looking into. So diabetes is a serious chronic disease. So it's, as of yet, there is no cure. And it has an increasing prevalence. So in 2001, it was estimated that there was about 10% that had diabetes. In 2030, there will be 100 million more people living with diabetes, and it's projected that by 2045, 1 in 8 will be living with diabetes. And type 2 diabetes is associated with multiple comorbidities and increased mortality. So even though the mortality difference between people with living with diabetes and non-diabetics the difference has shortened, then as of now, we still see that people on average live 8 years shorter if they have diabetes, and also have a notably increased risk of cardiovascular disease and also chronic kidney diseases. Then Novo Nordisk is also very prominent in treating people living with obesity, which is also categorized as a serious chronic disease where there is a large unmet medical need. Until recently, there was very limited, very effective treatments for obesity, and it was maybe also rarely recognized as a disease, but something that you could take care of yourself. As you can see here, then there is expected to be a 53% increase in people living with obesity from 2020 to 2030, estimated. So across the globe, it's more than 1 billion people. There is increased risk of metabolic disorders, so for example, diabetes, cardiovascular, but also mechanical, so basically the CV system in the outer limbs. And we can also see that there is reduced risk of aging the higher your BMI becomes. And the last therapy area which touch upon is cardiovascular and other treatments. So these graphics show that there is a strong overlap between obesity, type 2 diabetes, ACVSD, which is a type of cardiovascular disease, and heart failure. And it is still one of the most prominent causes of death globally. And about 30% of all deaths are caused by CVD. MASH, which was until recently not called MASH, but NASH, non-alcoholic fatty liver diseases, because it wasn't really seen until about 70 years ago. But now more than 250 million people are affected by MASH. And until a couple of years ago, there was no effective treatment at all for MASH. There's almost 1 billion people living with kidney disease, which, as you can can result in the need for dialysis and ultimately kidney transplants. And ACVSD is also a very prominent disease. And even though you can say that it's a very established therapy area and with many treatments, then there is still a very significant and growing healthcare burden coming from cardiovascular disease. So the possibility of reducing the need for surgical support with pharmaceuticals could really qualify as a reduction for the burden on healthcare. And as you can see here, the US annual economic burden of CVD is estimated to exceed $1 trillion in 2030s, which is not sustainable. So we need to come up with better ways of treating and hopefully preventing cardiovascular diseases. Yeah, so that was a quick run through what the therapy areas and what Novo Nordisk is. So next I will go into how we work with real world evidence and artificial intelligence. Yes, so the challenge, why do we enter into these spaces, is that, as you know better than anybody, then there is significant scientific progress in evidence generation and knowledge insights generation. And it's basically outpacing traditional drug development methods, which is based on how you would do it maybe in the 1960s. And novel ways of generating evidence really holds a promise for faster evidence generation, which would mean faster treatments to patients, lower cost, and faster access to the treatments. And basically, if it's not a viable business case, you could say, then the innovation goes away. So we see this in many infectious diseases where the business case is poor, which means that there is very little innovation in the area. Or you could take something like multi-resistant bacteria, which is really difficult area to get new treatments approved in a viable business case, which means that there is basically no innovation in an area that would really benefit from seeing novel treatments. So traditional evidence generation processes struggle to keep up with the scientific advancements and basically adopting them. But they do hold they remain undeveloped for sort of mainstream use. And sometimes, because I also have a collaboration with Berkeley, then I have this idea that you have people sitting in the ivory tower, develop methods further, and further. And then you have us on the outside really struggling to keep up and figuring out how can we adopt them to generate robust evidence, because there really is a need to make sure that this works across the entire patient population we are investigating, and not just a really good use case of n = 1. But if we do not find out different ways of generating the evidence, it will slow innovation, it will raise cost, and it will ultimately delay novel treatments for patients. So the promise with real-world data, which is already out there, because one of the most costly things for drug development is generating the necessary data to take the decisions. And AI is that it can improve efficiency and execution. And we believe that scientific progress centered in broad data science can improve the entire evidence generation process, which would make development programs way more efficient. And currently, development programs cost many billions of dollars to get a treatment to market. It could enhance evidence generation. So it is vital, of course, that we are sure of the safety and efficacy of the drugs we bring to the market. So we must be certain that what we are generating is robust and actionable, so that the doctors can take these treatments and give them to their patients and be sure that once it's approved, it does work for almost all. And it can be, faster and more cost-effective drug development, so we can get affordable drugs, and it would reduce timelines and resources, and we can work better maybe. And by using real-world data, we can also maybe make trial designs that are relevant to where people actually, sort of the natural setting that people live in and get their healthcare providers from. As you can imagine, then the clinical trial setting is somewhat artificial, where you have a lot more attendance to the patients and a lot more time with the healthcare professionals than you would get in a normal healthcare professional interaction. So the areas I chose to talk about today, well, first of all, I would say that a lot of it is based on academic collaboration. So we partner, Novo Nordisk partner with academic institutions. It's based in our DNA, as I said in the beginning, University of Toronto, University of Copenhagen for insulin and GLP-1, but really what novel methods out there would significantly help us in what we are trying to do. Then, of course, we work with AI to improve how we work. So there's one thing about how we do our everyday work and how we work with data and whatnot. But then there is the deliverables that we can. So can we use AI for efficient information and decision making? So we, of course, also looking into LLMs for rapid information about our market products, and could we use it for efficient safety assessment? And can we use it, of course, to shorten the time at drug development? And as you maybe have heard, because there's been a lot of buzz about it, is that one of the things that takes a long time in research is protein prediction. So I have a slide on that. And then because I know that Ahmed works a lot with images, I thought I should also take something about how we work with images. But we believe that if we could leverage image data further, then maybe we could have easier diagnostics. So for now, if you were to diagnose whether or not a patient has liver disease, then you have to take a biopsy, which is, aside from being very uncomfortable to get a bite of your liver, then it's also a rather risky endeavor, because there is a lot of blood in the liver and you will only get one bite. And maybe that's not exactly where the disease is most prominent. It was actually 1 centimeter to the left of where you take the bite. And then we have the real world evidence, which is about being able to use the data that's already generated in the existing healthcare without having to generate it ourselves. So first, a little bit about AI. And as you can imagine, we have an enormous amount of data. So we take a lot of omics, we take proteomics, we take genomics, we take sociomics, we take microbiomics, we have social media data, we have the clinical data, we have data from wearables, we have data from devices, we have data from imaging, And we also have data on how people behave. And all of this can inform us about the diseases, how they affect, how does high inflammation affect the brain, how does our molecules affect the body, what does it do when it goes in, where is it, which hormones is it that it increases or reduces, what is it that it removes from the blood. All of these data can inform us, and we can for sure analyze them better than we're doing at present. So we believe that AI brings innovation across the entire Novo Nordisk value chain. So if we go to research, where they do target discovery, they knowledge mine all the different protein structures and genetics of people. We could use robotics to decrease design cycles. We can do predictive pharmacology. So if we chain this receptor on some protein, what will that do in the body, instead of always having to sort of design the protein and look at how it acts in a petri dish and then look at how it acts in a mouse or a rodent. In clinical development, we can increase understanding of the patients. We can optimize our trial designs, how we choose the sites. We can forecast how our trials will behave, and we can maybe also do some pattern recognition. In product supply, we can, of course, optimize the way we produce our drugs. We can have better inspections, and we can have AI augmented process analytics. And likewise, there is plenty of areas where we can use it in commercial. So I was recently in San Francisco because we just bought this new computer from NVIDIA. Because one thing for sure is that in order to do all these things, we need a bunch of compute power. So we bought one of these supercomputers from NVIDIA, and he was just in last month here in Denmark to, as you can see, put the cables in and make it work. So we do try to make sure that we can actually do the things we want to do. And now they're building it, and we are trying to process some of our data through it. And this is about how one of the things that can take years and years is to figure out what does this protein look like? Can we design it so that it hatches better onto the receptors in the body? And if we can do that, we could essentially shorten the development timelines from maybe 10 years to 8 years, because that can take years to figure out what does a protein actually look like. And the purpose is also maybe to reduce the failure in clinical phases. So if we didn't predict it quite right, then maybe it doesn't do what we thought it would do. And then we start these lengthy, expensive trials that ultimately fail because it didn't hatch onto the receptor in the body that we wanted, how we wanted it to hatch on. And then there's something about how we use AI for the way we work. So we write a huge number of protocols and hundreds of pages. We write clinical reports, we write submission documents, and a lot of them are actually quite similar. And we have rules for how we assess the data they are based on. So we have built this thing we call Novo Scribe, which basically takes all the data output that we generate, and then it generates its own report that we can just read afterwards. And ultimately, it actually means that we can produce these documents 70% faster than we used to. And it can, it really changes how we work. And also these reports are not that much fun to write, because basically we say, if the table on A1C reductions or the table on weight reduction look like this or that, then there is a requirement that then you are supposed to assess it in a certain way. So there is no really intellectual power needed to really write these reports. Yeah, and then of course, as everybody else, we have also built some chatbots. So there is one in product supply where you can just ask it questions and it can answer, and it has this very sort of simplistic interface. But we are also building, building them elsewhere. So the next one I want to talk about is that we have a lot of information about our products, but we don't always remember all of it, or all the people across the organization don't remember all of it. So then you have to, somebody asks you a question about what's the effect of this or that drug in this or that population, and you don't exactly remember. So you have to remember, oh, that was that paper from 2022 and we're publishing New Integrative Medicine, and I have to look it up and then I can answer. So couldn't we maybe have our own internal chatbot that could just tell us what it looks like? There is, of course, a bunch of limitations. If we just use ChatGPT out there, then maybe it's not trained on exactly the data we want it to be trained on. It can also sometimes produce answers that are not 100% accurate because maybe it doesn't know and then it just really wants to answer. And also, it's not really trained in getting medical advice. So we tried to build one internally that could help us with this very real problem, where we did this retrieval augmented generation. So basically, we fed it, you can only know these documents. These are the only ones you can answer based on. We also told it that if you don't know the answer, then you have to say, I don't know the answer. And there was also a guide to create better answers. So what we served it was selected publications that we said, these are the publications you can know from. And then we scraped from public sites, sort of trial information from clinicaltrials.gov. We took protocols and analysis plans and submission documents. And then we trained it on a whole bunch of answers that our medical colleagues across the world often gets. And then the answers that they have said that written down, that these are the answers that they give. So we had a lot of sort of ground truth that we could serve it and train it on. And then we evaluated it, and we could do a quantitative evaluation on the ground truth. So we had this large question answer document and just see how accurate are you? And it also had to give references from which paper did it take the information from. And then we could sort of check the completeness of the answer. And of course, the metrics doesn't really give the full answer because, the answer can be correct even though a few words are flipped around. So we are fortunate to have a bunch of medical colleagues who could also qualitatively assess the feedback. Our colleagues are crazy about this little thing, but we do have some challenges because, as you may know, then when you read clinical papers, then the information is not all captured in text. A lot of it is captured in images and figures and graphics. And so we lost a lot of information. So the answers that we had in the ground truth documents didn't really correspond to the answers the little LLM could come up with. And also, we don't really have a user interface, so we need to build that. And we really want to have some graphical readings in there. And we are working on a solution, and because even though we have that big computer standing there, then maybe this is not exactly what it's to be used for. But it's not the only, these two are not the only chatbots we have. We have others, but this is to say that we, of course, also develop these along the way. Yeah, and then on to imaging. And imaging is actually quite big with us. I mainly work in development, but I did take a little one from research because of course we also use imaging analysis in the preclinical setting, where we look at genetic and chemical perturbations, we look at the microscopic imaging, and we analyze for, does it look like it should, does it move the way it should, and downstream we can make sure how the drugs are absorbed in the different liquids that we put it into. In development, we basically think that we would know more about a patient if we had some images, and we would be better at figuring out who to treat and when to treat. So this example is MRI scans of 7 individuals from UK Biobank, which is a big database on 500,000 people from the UK. And all of these people have the same BMI, about 27, and they have approximately the same waist circumferences, which is 91 centimeters, which are sort of the traditional, easy, accessible biomarkers you use to sort of see if a person has a healthy BMI or has a healthy body composition. But as you can maybe also see, then the body composition is very different in these 7 people, and especially the fat distribution is very different. And what is measured here is a different kind of where the fat is. And the first guy, with 2.4, is way healthier than the last guy with 9.3. So we hope that we could get a lot of biological insights using analysis of these images. And that way, it would allow us to get diagnostic, prognostics, and predictive biomarkers that would help the medical community. So basically, it's the diagnostics, who to treat. It's also the prognostics, is the treatment actually effective? Does it do what we want it to do? And does this disease state improve? So predictive biomarkers. And what we have worked on a lot is in liver disease, because as I told you, that the sort of golden rule for diagnosing whether or not you have MASH at present is to take these high-risk liver biopsies, which is expensive, it's risky, and it's very uncomfortable. So can we somehow just take an image of the liver and maybe in that way know it, because we can't really use clinical biomarkers, so we can't really measure it just from taking a blood sample. And we would, if we could really sort of diagnose it non-invasively, we would know who to treat and when. And the image here you just see is different stages of liver disease. So with the first one, you have a healthy liver, and then you have one where there's too much fat. When the BMI becomes too high, the body doesn't know what to do with the fat, so it starts distributing in the brain and the heart and the liver where it's not supposed to be. And then it gets fibrosis, so it becomes too stiff, it stops its functioning, and then that progresses more and more. You get cirrhosis, and in the end you get cancer, and then you have to get another liver. And this is the alternative. Your liver sits there in the body, and then you take a big needle and take a bite. And what we have worked on is MRI scans again, where we have a healthy liver, one with early stages of disease and one with advanced fibrosis. And then we have tried to sort of figure out if we, based on these images, could see what stage the liver is in. And it's actually turning out to be pretty good, almost as good as liver histology, because as I mentioned a couple of times now, liver biopsies are not that golden as you would think. And especially because liver doctors are often in disagreement how diseased the liver is. So this is turning out to be quite promising. And then we also have some in production, but I will skip that. And then there is also something about sort of explaining what it is that you see. So we also tap into this explainable AI. So in traditional sense, you have some training data, then you have a learning process, you have the trained model, and then you have evaluation of the output. But this black box setting is really unattractive to most doctors. So can we somehow tell the doctor, why do we make this assessment? And one possibility is saliency maps. And it has some success in, for example, when you use fundus or retinal images of the eye, where you can see here is the original image of the eye. And then there is saliency maps with these green dots where it can tell you, what did I look at in order to assess the age, the gender, the systolic blood pressure, and whether or not this person was a smoker. And this is actually some of the really useful things of images, that it can predict the age. And you can, of course, say there is easier way to get a person's age by instead of taking an image of the eye, but it's at least one of the things that it does really, really well. And you can also say there is easier way of assessing a person's gender than taking an image of the eye. But what is biologically interesting is that the model is actually really good at figuring out whether it's a male or a female based on these images, whereas a eye doctor wouldn't be able to do that. So the model sees something that an eye doctor doesn't usually see, and you can see it's very much focused on the OCT scans of the fundus. And yeah, systolic blood pressure, that has been known for ages, that the eye is the window to the body, and looks, of course, at the veins, and then it can see whether there is damage from smoking. I do think that there's one very important thing not to forget when we talk about all these images. And I took this paper that was very recently published in October 31st in JAMA, that AI screening for diabetic retinopathy is actually almost not used, and which is maybe a little sort of offhead, because the number of publications about how you can use images of the eyes for different diseases and how well it performs, it's just, there is more publications per day than I would like to count, and the amount of work that goes into that. But on the sort of downside of that is that apparently it's not used. So maybe we are not solving the right problem. And this particular one is about, we're really good at assessing retinopathy in the eyes for people with diabetes, but nobody's using it. And is that because once you have diabetes, which you can diagnose without an image of the eye, the doctor already knows that there's a high risk of retinopathy, so it's not really necessary. So are we solving the right questions? Are we solving something that's useful in the clinic? Which is maybe a question we should remember to ask ourselves once in a while. Yeah, that was it for AI. And now I will quickly run through how we use real-world evidence in our development programs. So just to sort of capture again, development is all about making these trials. Active comparator trial, regulator says how long, what are the endpoints, who are the people that you get into the trial, how should you power it, and all of those. And everything is pre-specified. Who to treat, when, how, what to collect, and what do you do if the measure is large or small, and how do you analyze it. And a little bit about the drug development process. So the clinical testing is by far the most expensive thing that we do. It costs about 1/3 of the total R&D cost. And R&D usually amounts to about 15% of net sales. So for example, when we do a cardiovascular outcome trial, they are 2 to 5 years long. They have maybe 12,000 patients, and they cost about 5 billion DKK, which is maybe roughly $1 billion US. But we're looking into that they will take twice as long and need 8 times as many patients. So we definitely need to do something else. Yeah, and then I thought I would just give a little bit of a game changer here, but I am cognizant of time. But it's just to say that there is a history where We saw an increased risk of cardiovascular disease with diabetes drugs. So FDA said you need to address CV safety. They came out with a guidance saying you can't get a drug to market without addressing the safety of cardiovascular risk. And the governance of that was actually quite complicated. I will jump over that. But the impact really was that we saw over the years, every company had to start these huge trials in thousands and thousands of patients across the globe. And what ultimately happened was really there was a change in how you manage type 2 diabetes across the globe. So what happened was that all of a sudden you saw studies showing that not only do you not increase the risk of CV, but you can actually have a benefit with these drugs. So this is a result showing that you get a 15% benefit if you're treating with an SGLT2 or an GLP-1. And all of a sudden, instead of making it because you had to, you made it because you have to do it in order to get a good business case. So just in Novo Nordisk, we started trials now in 20 and 17,500 patients, and more are coming. The problem is that these trials, they are event-driven. So you never really know how long time it's going to take. You just know that you need to collect maybe 1,000 deaths. And they are very governed by regulatory for how they are to be conducted. I will jump over all of this. But the challenge is that if the trial looks maybe like this, then as time goes by, maybe it takes like 5 years, then people decide they don't want to be on treatment anymore. Maybe they want to come back on treatment. So you don't really know how much exposure they really had. Maybe they decide that they don't want to be part of the study anymore, so they drop out. You can die for other reasons than CV. You can start taking other drugs. You can stop taking other drugs. You can have something like COVID happens where all of a sudden you can't see your patients. And basically what happens is that the way you wanted to analyze your data is no longer possible because you didn't get the data that you wanted to. And also you can imagine that the benefit of the drug depends on who's asking the question. So you can have one that says, if I give you this drug, how much lower will your risk be compared to if you didn't get the drug? So that's just meaning that you get the prescription. That's the regulatory interest. But a patient says, if you take this drug as you're supposed to do, what happens then? And the physician might also want to know, if you take this drug, but you also use this other drug, what happens? Or you could say, what would this outcome of this study have looked like in a world that wasn't affected by COVID? Yeah, and I will skip this. So basically, we have challenges. There is, we don't get the right representation in our trials. The trials are getting more and more expensive, which means that it's harder and harder to bring a new drug to the market. And it's really difficult to analyze them. But there is a promise. There's a promise of maybe integrating it with the real world data that exists and making sort of a bridge between the data comes from the RCT and the data comes from the real world. And we also have this idea that maybe we can do federated learning across. All of this really requires novel ways of analyzing and using the data, methods that are in development. So I will skip all of this and just say that we believe that data science is really relevant across all aspects of development, from early target discovery and all the way out to how can we benefit population health. We just need to figure out how do we use these novel scientific advances. Yeah, that was it. Thank you. I made it in time.
Thank you. Any questions in the room? So I had a question about real world evidence. And
Yes.
I'm not sure what's like if you take the example of the GLP-1 therapies and people are showing every day that they work for like new indications other than obesity and diabetes. So how do you decide what the outcomes were like as a pharmaceutical company that is already like discovering and commercializing that drug? How do you think about the endpoints, both from the efficacy and safety perspective, when you are designing a trial, especially for drugs that seem to have the potential of affecting so many different outcomes? And like, what's your incentive engaging with real-world evidence analysis? And like, how do you get the data? Because in the US, like academic institutions own the data, but I guess maybe in Denmark, there's like there's more data sharing or more like national data sets.
Yeah, so that was a bunch of questions. So usually we have medical colleagues with huge clinical insights. So if you see that we measure so many things in the patients, and sometimes you will see, oh, there is actually an improvement in these liver biomarkers. Maybe that does something with liver disease. Then we engage with regulatory to say, if we believe our drug could have a benefit on liver disease, what kind of endpoints would be interesting to you? And then FDA has a very specific guidance. If you want to have a claim that you can treat liver disease, then you need to show these and that markers. So if you want, for example, a CV claim, you need to show that you lower heart outcomes, which is dying from cardiovascular reasons, getting an MI, myocardial infarction, or getting a stroke. And those are the endpoints you need to show that you lower, and otherwise you don't get a claim. And if you don't get a claim, you can't talk about it. Then you can publish something that you think that this happens, but it's only in the medical scientific community you can talk about it. You can't sort of market it.
Yeah.
But so it's of course a scientific progress where you see there is potential these and that effects. As opposed to data, then there is a multitude of data vendors across the world. So in the US, Novo and other companies buy access to claims data that's collected. So there is Optum and Komodo and many others that sell us data that we are then allowed to look into. And in Denmark, there is national health registries that we can get access to through academic collaborations. In UK, you can get access to their national health registries by buying access. So there is usually a way. Of course, everything has to be approved. You can't just go explore. You have to come up with a protocol. I would like to look at this. They assess the protocol, and then maybe they say, yes, you can investigate this in our patients.
I see. Thank you. I have a couple of questions in the chat. What are the best ways to partner to get real-world data for federated learning? It requires HIPAA protected ML solutions, which doesn't exist yet.
Yes. So the issue that I see from federated learning is that where federated learning could help us is that we work as a global company, so we want our drugs available in the US and in Germany and in Japan. But we can't take the data in US and put it in our database together with the data from Germany and the data from Japan. So we could make an independent analysis in Japan and one in Germany and one in the US, but then of course we lack power and we can't really transform the insights from one database to the other. And we might have reason to believe that sort of the mode of action is not different across nationalities. So we believe that there could be an increased efficiency by tapping into federated learning theories. And we have written one paper with Mark on this, but we haven't really sort of commercialized it yet, but we have high hopes.
Yeah. Another question regarding the image-based models that you mentioned. So you talked about machine learning applications that look like decision support systems, like the diabetic retinopathy application, for instance. And I wonder if Novo, like what would make Novo interested in this kind of application? Because it doesn't seem to be related to drugs.
So usually there is sort of, FDA has qualified 7 different biomarkers. And right now we are looking very much at diagnostic biomarkers. For example, for liver disease or for heart failure, you would like to be able to differentiate between if you have preserved ejection fraction or preserved or reduced ejection fraction. So can the images help us with those diagnostics? And then it's diagnostics. But then there is also prognostic biomarkers, which is more to say that can we see if we give this treatment, can we then have a sort of mode of action image of what happens, where did it improve something? Because maybe we can't see it in a blood sample, but we could maybe see it on an image. So it depends on what kind of biomarker you're looking for. But it would be quite expensive for the healthcare systems if you had to use an MRI scan to see if you're, for example, let's say, if you get a hypertension drug, then you can see if your blood pressure is going down. But imagine you had to go into an MRI scanner every time to see if your drug did some benefit. So it's not really, yeah.
Other questions in the room? Paula?
Last thing you mentioned, like that, why are trials getting more expensive? Like, is that adjusted for inflation, or like what is making the process more expensive over time? Because I was thinking maybe develop new methods so that, as you said, like the idea was that we make it faster.
I think I need to have the question repeated because the sound was not.
Yeah, the owl never works. So I think the question was, why do the costs of clinical trials increase over time? And Paula's question was like, it seems like we are developing methods or like procedures to make things more efficient, but it seems that the prices are going up. So is there a reason why that's the case?
One of the reasons why the price is going up is because that for a lot of the indications we're looking at, there is already something there, and overall the longevity and the mobility of the population has improved. So if you look for the rate of CV in the 50s, it was much higher than it is now. So if the rate of dying from cardiovascular disease is going from 10% per 100 patient up year to 3%, then of course you just need to increase your trial a lot. So one thing is that it's a good thing that people are not experiencing these events so much, or you are adding on a therapy. So instead of going up against placebo, you're going up against an approved drug that just does it better, which again means that you need more people and longer time. But the drugs we have are still not good enough. So you still want better treatments and better therapies. And then, of course, there is the whole sort of the number of safety assessments you have to go into the requirements from regulatory is also going up, what you have to measure. So if you could maybe sometimes you just measured, your age and gender. Now you also have to take a proteomics profile and a genetics profile, and you get to have that sequenced, and that increases the prices a lot.
I see. Yeah.
Can you speak about what the main costs of a clinical trial are, and specifically wondering if like testing, monitoring the biomarkers of these patients on a higher frequency is a big factor in the cost?
Yeah, not sure if you heard the question.
It was something about increased costs.
So like I think we was asking about the breakdown of like what's the most costly aspect of a clinical trial, and whether repeated testing is what you're asking about.
Yeah, like biomarker.
Like collection of biomarkers, and like is that a big factor?
Yeah. So of course, if you could just have a person come into some sites and answer some questionnaires, that would be a lot cheaper than if you had to have him come in and have a liver biopsy or an MRI scan or a DEXA scan. So the more sort of invasive and costly the testing of the humans are, that will drive prices up. And then if you have to get 20,000 people through it, and they have to come every month for a year, that just drives prices up again. So it's a combination of how many people do you have to have come by, how many sites do you have to have, what kind of information do you have to capture on the people, and things like imaging and surgical procedures and omics profiling is very costly.
Okay, maybe one final very quick question. I know it's very late on your end. Sorry to keep you. I think it's 8 p.m. on your end.
It's okay. It's nice
So
to have some questions.
Yeah, so one final question about the regulatory aspect of AI models. If you have thoughts on this, or you can just briefly tell us how the FDA and the European agencies are thinking about this.
Yes, so they're thinking about it a lot, but they haven't sort of finalized what they mean. But I can say that I was at a conference in San Diego this summer where the commissioner from FDA, Bob Califf, was present. And there was a lot of sort of tech companies saying that FDA was halting innovation to reach the patients because the regulatory pathways were so expensive and cumbersome. And he replied in a way that I thought was really, really common sense, that they were there to protect the safety of the patients. So they have to make sure that whatever gets to the patients does what it's supposed to do and is safe to use, which means that ultimately you have to run it through testing in human beings. And that takes time. And how are you going to, if you bring a protein, then you know that that protein is going to look like this today, tomorrow, and in 10 years. But if you bring an AI model, like, are you going to lock it? So I mean, we all know that it can change and it can learn. So what's the regulatory pathway for bringing these things to people? There is very limited AI being approved, or very limited interventions based on AI being approved. For one thing, because we haven't really seen the big efficiency gains. So the number, for example, you can imagine that, oh, I'm enrolling in this or that program, and it looks really great for the people who starts in this program and get some interactive advice. But usually after a year, it's only 40% who are still there, and those 40% might have benefited anyway. So if you start imputing the 60% that didn't show up after 12 months, the benefit goes away. So there really hasn't been that much that really showed a robust in effect long term.
Okay, thank you so much again.
You're welcome. Thank you for having me.
Thank you, and thanks everyone for attending, and see you next week.